Today, I'm going to present something which I did as part of my PhD thesis work. And while I'm working at Schlumberger, before this, I defended my PhD last year in May at Lillehoj Research Group at Mechanical Engineering, Rice University. So, as Dr. Cindy mentioned, this research was published in Microsystems and Nanoengineering. If someone is interested, you can scan the QR code and it'll take you to the page. So now let's talk about the motivation for the research topic which I'm presenting today. So in this research, we wanted to quantify CD4 positive cells. They are clinically relevant cells in the sense that they kind of tell the status of immune status of anyone, and then it's also very critical in when it's HIV related diagnostics. So in conventional method, these CD4 positive cell counts are done in flow cytometry, using flow cytometry, which is bulky, expensive, and kind of needs a long preparation time, and always needs someone who knows how to use this machine, a very like a trained staff to run this. So our question was, can we speed up and simplify conventional detection without sacrificing the accuracy? Then we looked into literature and we figured out there were a lot of microfluidics research which was done to quantify CD4 positive cells. But what we realized after reading most of the research papers that most of them, a few of them employed complex microfabrication techniques, which kind of makes the device expensive because you have to pattern electrodes and other things. And then some of the research were involved sample preparation, example lysing the RBCs, because they are the 97, they almost have more than 97% of the cells present in blood. And some of them also included complicated immobilizing protocols, like immobilizing protein on a chip, on a surface. And then obviously at the end, there was always a complex readout method, which all again needed a big machine to solve this issue. So these were the limitations which kind of we saw in the literature. So we thought, why can't we just integrate microfluidics for the flow and convolutional neural network for optical detection and quantification of the cells? So which would enable rapid quantification of cell types in human blood. Before before I go ahead, I wanted to explain the basic principle behind the flow, because this research we wanted to eliminate most of the peripheral equipments which kind of don't have a primary purpose to be there. They are just there to kind of support the main system. So one of them was the pump. And in order to explain how we deleted the pump from the system, we have to understand how we used a slug flow to come up with an idea which would delete the pump. So imagine you have a capillary and you have sample in the capillary and you have like your fingertip on top of it. The flow, the slug, the volume of fluid inside the capillary won't fall. As soon as you lift your finger, the slug would start falling. That's slug, falling slug. So what happens when a slug is inside the capillary and when you lift your finger? It is acted upon by three forces, which is viscous force, because it's moving, it's falling down. It has surface tension force on the meniscus top and bottom. And it also is, and there is force, gravity, it's everywhere on Earth. So the combination of all these three forces actually makes slug fall at a steady pace, at a constant pace. So this was the basic premise of the principle behind how we could achieve a pump-free operation. So given the principal idea, we came up with a device. What we did is we created a microfluidic device which had 1 millimeter cross-sectional, two reservoir, 1 millimeter square cross-sectional on top and bottom, connected through a constriction. If you could see, if you could see here that there is a constriction which is connecting the inlet reservoir and the outlet reservoir. So what the size of the reservoir is the top and bottom reservoir are exactly identical in cross section. That is 1 millimeter side, but the constriction is 30 microns. So overall, what happens is when we the meniscus and top and bottom meniscus always stays in the same cross-sectional area, so there is no cross-sectional area change, so the surface tension force doesn't change. But this cross-section, the constriction provides enough drag force on the slug that we could control the flow of we can manipulate the flow or the fall of the slug inside the channel. After doing this, what we did is we tested our 10% diluted, 10x diluted blood in our chip, and we saw that actually, if you could see the plots on the right, you would see that the meniscus position is quite linear. So the fall, as we have predicted by theory, it is constant. The velocity of fall is constant. And we tried manipulating the flow rate by changing the thickness of the cross constriction. So if you could, by changing the length of the constriction length. So because the constriction thickness is 30 microns that we kind of kept fixed, if we increase the length of the constriction, we can change the flow rate. So if you could see the second plot, you would see that as we increase the constriction length, the drag force goes up and the fall slows down. So the fabrication of this device was done in our lab. What we did is we designed this. We had the design and a CAD software. We created a CNC code. We generated the master mold using a micro CNC, and then we cured PDMS on top of it, and then did the air plasma bonding. And then there was a step which we added to the whole process is heat treatment for contact angle recovery, which I will try to explain later on slides. And after the bonding, when we do the plasma bonding, we have the device. So why did we do the heat treatment process is to understand, we have to understand the surface wettability in our device. When we make any device using plasma bonding, the surface is hydrophilic in nature. So for our operation, if you look at from a surface wettability perspective, we want the slug to fall as a single body. While if the surface is hydrophilic, it would start falling through the corners of the channel, because channel is square in cross section, and that would change the whole the assumption that we wanted the slug to fall at a steady pace. If the slug starts to fall through corners, the flow would be non-uniform. So in order to understand this, the theoretical background is that the contact angle, the surface should be hydrophilic in nature. In order to achieve that, we did the heat treatment process. So when we keep a chip, a fabricated chip at an elevated temperature, the contact angle recovery is very fast. Fast in the sense it is in days instead of having in weeks. So we kept the device at 90 degrees Celsius for 48 hours. And in this image, you could see that just after, if a PDMS is treated with the plasma, the diluted blood kind of makes a very acute angle, 17 degrees. And after the recovery of the contact angle after 48 hours of heat treatment, it kind of recovers the hydrophobicity which it possesses. The PDMS is supposed to possess. So after this, we did a long-term storage test that if we keep this device for very long, would the recovery, would it go in backward direction or anything would happen? Just by keeping in a normal ambient condition in a closed box, we observed that there was no change in the contact angle over 30 days. So the device is fine and can be kept for long durations and has a longer shelf life. As I said, we wanted to quantify cells using convolutional neural network. It's an optical based technique. We had to somehow tag the cells because these CD4 positive cells, they don't look distinct if you look it under the microscope unless you stain them or they have some kind of a visual cue on them. So what we did in this research is we incubated this CD4 positive cells with magnetic beads. Magnetic in the sense because these beads are right now, they are available in magnetic form, but we can also have beads which are non-magnetic in nature. So these beads are covered with monoclonal antibodies, which makes them highly specific. And when you have micron-sized beads, like 4.5 micron sized beads, you give immense surface area, which increases the sensitivity of the process. So you could tag most of the cells in the sample very quickly when they are in bead form instead of being on a surface. So here are few images where you could see that the CD4 cells look very distinct when they have beads on their surface. On the first image, you could see CD4 cells. It may have more CD4 characteristics, the protein specific protein on surface of it, so it has more beads bound to it. While on the second image, you could see the CD4 cells has 3 or 4 beads on their surfaces. So if you could look this image, a human eye can tell that these cells look very distinct when they are attached to bead. So if a human can tell if it's distinct from its background, AI can also be trained to quantify these cells in the sample. So now the second challenge was because it's an optical based technique and the gap size is only 30 microns, we didn't wanted the cells to overlap. So majority of the cells in blood are RBCs, and you could see in the first image the undiluted blood where RBCs are all over the place. We cannot identify because the CD4 cells which is tagged with bead would be on the other side of the RBCs or be like totally opaque or hidden behind them. So we tried doing dilution, and then we figured out at the optimal dilution was 10x dilution, because as there are two, it's an optimization problem where if you keep on diluting the sample, what happens is the volume that you need to process to get us statistically significant answer would also increase. You have to process more and more sample. But if you don't dilute it enough, then you'll have more RBCs and detection becomes very tough. So in total, we decided that 10x diluted blood was 10x dilution was the optimum dilution for our samples. Now we have cells which are tagged with beads. We have samples, so we took a lot of images. We captured a lot of images. We trained a CNN algorithm. When I started this research, YOLO V5 was one of the fastest inference-based inference speed. So we tested YOLO V5, and we almost had 4,500 images. We split that into 80-20 testing and training. Training and testing set. We trained the algorithm and on Google Colab free T4 GPU, and then we use this model to do the inference. And the inference was because in every image we take when the cell flows in front of the through the device, we know the exact volume and we know how much area we are looking at. So we could quantify how many cells and how much volume is there. So we could quantify the concentration of cells in the sample. So the workflow for quantification is that we hit finger prick, we take blood, we dilute the blood, we incubate it with CD4 anti-CD4 positive beads with it, and then we dilute the sample to 10x. We feed that with 20 microliter of the sample. Almost the volume need not to be precise, but it has to be close to 20 microliters. It is fed into the chip and allowed to and then in a slug format, it would fall inside the microfluidic chip which we fabricated. It is placed in a way that the microscope can actually record a video of samples flowing through, and the camera, a camera which is inbuilt in the microscope would take images at 15 fps, a video at 15 frames per second, which we would post-process using the convolution neural network, which will give us the count. So the overall time period to time scale to do this is that for the incubation first step, we only needed 5 minutes because they are micron size beads. 3 minutes for the flow, for 20 microliter of sample to flow slug to fall through the chip, and then 5 minutes for post-processing. I just wanted to tell you that the 3-minute flow timing and the 5-minute post-processing time can be overlapped because they are independent, and as soon as it could be a live inference. So here is a video of the sample. Yeah. So here is, it's a 10x video, and you could see that's the constriction region, which kind of disappears when it is a video, but you could see there's blood on top the inlet chamber, flows through the constriction and comes in the outlet chamber, and it falls, and in the constriction region the camera actually, and you can see this is a video which was which was post processed using the convolutional neural network, and you can see the algorithm identifying the CD4 positive cells in the flow. It's highlighted with the orange square around it. And after this device was ready, we did some of characterization measurements, and then we finally did the final test on 8 blood samples from volunteers. These were all healthy samples, and we tested these samples against the gold standard, which is the flow cytometry, and we saw that the results are within the 10% deviation, if you could see. And the Band-Altman analysis suggested that there is slight positive bias of 1 cell per microliter. That means the system is not biased towards one side. It's almost it's not biased toward one side, and the results are randomly, the error is randomly distributed on both sides of the mean. So the conclusion of this study is that we built a device, a microfluidic AI-enabled microfluidic cytometer, which could rapid in less than 15 minutes could quantify immune cells. And the operation of this device is pump-free for sample transport, which totally eliminates the use of a microfluidic pump in the sample. The CD4 positive counts were actually then compared with the gold standard flow cytometer, and then we observed that in all the 8 blood samples we tested, the deviation was less than 10%. Overall, this device, the whole protocol and the whole device is 4x faster. The protocol is simpler because it doesn't need lysing of RBCs, and the cost is also the cost effective because it's only a finger prick blood, like only very few microliters of blood incubated with beads, which it's very small volume of beads that is used. Moreover, this technology can be adapted to different kind of cells. CD4 positive was just a model target in this study. For this acknowledgement are my co-authors, Tyler and Dr. McHugh, and Dr. Lillehoj, who is also in this talk today, in the webinar today, and then shared equipment facility, which I took a lot of advantage of at Rice, and the funding which we had, and my research group, obviously.